what I want to do in my talk this morning is take you through Imatinib as a paradigm of targeted cancer therapies and why it was successful, what we've learned from our clinical trials, and then how we expand that to other diseases. And oftentimes I actually use a video just to help get you into the understanding and recognition that as I talk about CML, what I'm really talking about is a paradigm for how we can do this across many, many different diseases. And I hope you can understand that by understanding what I'm talking about with one specific disease. So when I think about translational research, to me, the heart and soul of it is you start by describing a clinical entity, you then understand its molecular pathogenesis, and then you design a therapy based on your understanding of the molecular pathogenesis. Now, in the case of CML, that took about 160 years, and I suspect we can go a little bit more quickly in this day and age. But this is really the heart of what I think about translational research. But when I got into this field, I thought it was this one-way translation from lab to the clinic, and that's where it ended. But one of the things I've learned through the Imatinib clinical trials is it becomes a translational research cycle. And that as we take our drugs into the clinic, we also need to think about how our drugs are working, why they're failing, go back to the lab, and then complete a translational research cycle. And I'll give you an example of that during my talk. So let's start with clinical description of chronic myeloid leukemia. 1845, two pathologists, Bennett on the top left, Virchow on the bottom right, described cases of chronic myeloid leukemia within 6 weeks of one another. Now imagine 1845. There were no computers. There, in fact, were no typewriters. These articles were typeset by hand. There was no postal service. So these were delivered by, I'm not even sure how. But nonetheless, in 1845, there ensued a vigorous debate about who was really first. You see, that's not new in medicine either. Now, ultimately, Virchow capitulated that Bennett had described his case indubitably 6 weeks prior to mine, and therefore Bennett gets credit for being first. I'll fast forward 150 years. We now know that CML makes up about 15% to 20% of all leukemias. The incidence is 1 to 2 per 100,000 per year, about 5,000 new cases per year. It can affect any age group. The average age of onset is about 50 to 60 years of age, and historically the survival was about 3 to 5 years. If we're going to do anything about this disease, we need to understand the molecular pathogenesis. So, 1960, again, two pathologists, Peter Knoll, David Hungerford, working in Philadelphia, described an abnormal chromosome in the blood and bone marrow of patients with CML. And this was the landmark paper they published in 1960 in Science, and this is the entire paper, 3 paragraphs. Immediately tells you that quality and quantity don't necessarily equate. Now, if you look at the last paragraph, which is where you can wax philosophical about the major implications of your finding, they said the findings suggest a causal relationship between the chromosomal abnormality observed and chronic granulocytic chronic myeloid leukemia. Now, in 1960, nobody believed them. They thought that this was an associated abnormality that had nothing to do with the molecular pathogenesis. It turned out they were quite a ways ahead of their time, and in fact were quite right about what they had said in that last paragraph. In 1973, Janet Rowley, working at the University of Chicago, showed that what was thought to be a short chromosome 22 and had been named the Philadelphia chromosome actually came about because of a chromosomal translocation between chromosomes 9 and 22. By the 1980s, when people mapped oncogenes to chromosome locations, it became clear that as a result of this chromosome translocation, the Abelson tyrosine kinase located on chromosome 9 had been translocated in its entirety to chromosome 22 and fused to a gene of unknown function called BCR for breakpoint cluster region. So in 1990, when I got into this field, this is what we had as a therapeutic target. BCR-ABL, product of the Philadelphia chromosome, present in every patient with CML. From animal studies done by George Daly and David Baltimore's lab, we put BCR-ABL into mice, they get leukemia. The first experiment I had done in collaboration with Jim Griffin, was to make a BCR-ABL inactive protein, and this had no transforming function. So it was clear that the kinase activity of the BCR-ABL protein was absolutely essential for all the transforming functions. So if you think about this schematically, BCR-ABL, a tyrosine kinase, binds ATP, transfers phosphate to tyrosine residues on substrate proteins, and that causes all the abnormalities we see in CML. So you could imagine if you could block binding of ATP to this specific protein, as shown on the right, you'd have a specific therapy for this disease. Now, it seems odd to think about kinase inhibitors as unusual, but I'm going to take you back to 1990, when kinases were considered not a viable target. And that's when I got into this field. And the problem was, I just showed you ATP. And the view was, ATP is ATP. There are 500 kinases in the kinome. If you block one, you'll block them all. You will never have specificity. The second was that they'll never work. And there were two problems for why they wouldn't work. Biochemists said ATP concentrations are 1 millimolar in cells, and you'll never have a competitive inhibitor that will work. And oncologists, being skeptical, their view was, when has a single agent in cancer history ever worked? So a single agent therapy just won't work. Why even bother? The third issue was, even if you began to work through some of those issues, they're going to be horribly toxic. And if you looked at some of the early knockout animals that had tyrosine kinase knockouts, PDGF receptor, embryonic lethal. EGF receptor, embryonic lethal. ABL null mice are not particularly viable. So even if you had specificity, the view was these kinases are so critical to cellular function that you'll have terrible toxicity. But the real death knell was the market size. 5,000 patients per year. The marketers from Novartis, their view was, we'll make $100 million a year at best because of the market size and market penetration. And if you do conservative estimates, they were probably right. So their view was that if it costs us $500 million to $1 billion to develop a drug and we get a return on investment of $100 million a year, it makes no sense to go into this market. Nonetheless, I began testing compounds in 1993. Nick Lydon from what was then Ciba-Geigy, now Novartis, sent me some compounds, and our lab showed that this one, which is now Imatinib or Gleevec, was the best compound that they had in their arsenal at specifically inhibiting the ABL kinase. So in '96, we published this paper in Nature Medicine, which showed we had an ABL and a PDGF receptor inhibitor, which is where it started. We also added KIT to its inhibitory profile. We could show selective killing of BCR-ABL expressing cells both in vitro and in animal models. And the chemists at Novartis were able to make this into a highly bioavailable oral formulation, which meant that we went to our clinical trials, we had a pill. So in our phase 1 clinical trials, within 6 months, we reached therapeutic doses of 300 milligrams and above. And in chronic phase patients who had failed standard therapy with interferon, we had a complete hematologic rate of 98% in a phase 1 trial. In blast crisis, which rarely responds to anything, including standard chemotherapy, we had close to a 60% response rate. I'll fast forward and skip all the phase 2 and registration, just go to a phase 3 trial, which treated newly diagnosed patients with Imatinib, and you can see that CML-related survival at 5 years is 95%. So remember, historically survival was 3 to 5 years. Now 5-year survival is close to 95%. But Imatinib's not perfect. At 5 years, about 7% of patients will progress to more advanced phases of the disease, and about 17% will have some evidence of disease progression, whether that be return of the Philadelphia chromosome in their bone marrows or loss of a complete hematologic response. Interestingly enough, the risk of relapse actually declines over time, with a peak in the 2nd and 3rd year and declining pretty much every year thereafter. What that means to me is that the resistant clones are probably present at the time we start treatment. We actually don't induce them with therapy, but we do a very good job of selecting for them on therapy. So now we've got to go back to the lab, and we've got to ask, why do some patients relapse? And we start with, we have an ABL inhibitor, and is the kinase inhibited? Because if the kinase is still inhibited when a patient relapses, it has to mean that something else is now driving their cancer. If the kinase is no longer inhibited when patients relapse, it has to mean that somehow the drug isn't hitting the target. Does it get pumped out of the cells? Does the target get amplified or mutated? Or is the drug metabolized? These are all questions we can answer. As it turns out, in the majority of patients who relapse, the kinase is no longer inhibited. And as we've learned, initially from Charles Sawyers and many others have contributed to this work, it's actually mutations in the target of imatinib, the ABL kinase, that gets mutated. And here what I'm showing you is a schematic of the ABL kinase from residues 240 to 500, which encompasses the kinase domain. And vertically are the frequency at which these different residues are mutated. So you can see mutations are scattered throughout the kinase domain. There's a high frequency at this one residue in the middle in red called 315, and others at the ATP binding loop where the drug binds. So we can actually learn a lot about how resistance works by looking at structural data from John Kurian. So here's this threonine 315. And on the left, you can see STI 571 in gray and threonine 315 in blue. And you can see that they actually contact one another. And in fact, they hydrogen bond at this residue. On the right, in sort of orange, is the mutation to isoleucine, which is a much bulkier residue. And you can see that there's a steric clash. There's a bit of that orange that sticks right up into where imatinib should bind. So clearly, if you have a direct binding site between imatinib and the ABL kinase, you'll get steric hindrance. Now, the other way that we get resistance has to do with how imatinib binds. And here, if you look at that red activation loop, you can see it has two conformations. There's the open conformation, and there's the closed conformation. Now, in that closed conformation, I've shown imatinib. So I'll come back here, and in purple is imatinib. And what imatinib does is it comes into the binding pocket and shuts this activation loop down in that off conformation. So now you could imagine we could design drugs to circumvent resistance. One way would be to look up at the top in blue, where we've got a drug that binds regardless of what that red activation loop does. That's what one company or two companies did, Pfizer and Bristol Myers Squibb, in designing a specific inhibitor. And the other way was what Novartis did, was to look at the binding of imatinib and say there's some mismatches in that activation loop. Let's do some chemical modifications and make a tighter binder. So that's what these companies did. They went into clinical trials with these, and we have Nilotinib, which was a structural modification of imatinib, and Dasatinib and Bosutinib, which are just structurally distinct inhibitors. Both are more potent than ABL inhibitors than imatinib. They still have one hole in their coverage, this 315I, because of the direct binding site. All of these have gone through clinical trials. All three of these drugs are FDA approved for imatinib resistance. They have significant activity in patients with imatinib resistance. They all have this hole in their coverage, 315I, and relapses, you might expect, are pretty common, now being driven into this 315I as one of the sole mediators of resistance. So to get around this one last hole in coverage, Arid Pharmaceuticals designed a 315I inhibitor. And if you look in orange and purple, you can see imatinib, and you see in purple that there's a little bit of imatinib sticking into this isoleucine at 315. What they designed in green was another compound that has a little bit more of a planar surface right at that isoleucine surface, so it can actually fit into the binding pocket. That drug is now at the FDA, and it has significant activity in patients with a 315I mutation. And in fact, it actually has significant activity in all the imatinib-resistant mutations. So where are we now a decade after imatinib approval? We have imatinib as a standard therapy for patients with CML, and it clearly significantly prolongs disease duration. We have relapses, mostly due to kinase domain mutations, and novel ABL inhibitors with significant activity against all the resistant mutations. But interestingly enough, we can't take patients off therapy. Most of our patients are doing well. They have well-controlled disease. But if you do PCR for BCR-ABL, they still remain molecularly positive. So in my view, the next quantum improvement will be eradication of leukemia stem cells. And if we could get to that paradigm, understanding how to get rid of cancer stem cells, we'll have the next quantum improvement in treatment. So I want to move on and talk about where else has Imatinib worked. Now, if you remember, the Imatinib targets were PDGF receptor, ABL, and KIT, and we have examples where Imatinib has worked in each one of these targets. So gastrointestinal stromal tumors, where most of them are driven by KIT mutations. 2 or 3% of melanomas have KIT mutations, and Imatinib has shown significant activity in both these diseases. Hypereosinophilic syndrome, driven by PDGF receptor rearrangements, and DFSP skin sarcoma, also driven by the PDGF receptor activation. Again, significant, rapid, dramatic responses in these 4 diseases. I'll talk for a couple of minutes about gastrointestinal stromal tumor because there's some instructive lessons here. Gastrointestinal stromal tumors used to be called intestinal leiomyosarcomas, but in fact they're pathologically and clinically distinct entities. Gastrointestinal stromal tumors comprise about 5,000 patients per year, and historically, if they responded to chemotherapy, that was worthy of a case report. In 1998, a Japanese group published that activating KIT mutations were present in the majority of patients with gastrointestinal stromal tumors, and as we began to see activity of Imatinib and CML, George Dimitri, Chuck Blanky, and I were able to convince Novartis to expand their clinical trials to patients with GIST. And what we saw was very similar to what we had seen in CML blast crisis, a chemotherapy refractory disease that now had a 54% response rate, and even patients with stable disease saw a dramatic survival benefit. In gastrointestinal stromal tumor, the patients often responded rapidly and dramatically. Here I'm showing you a PET scan. On the left, you can see massive uptake in tumor in the liver. And actually, within 24 to 48 hours, and here I'm showing you 1 month, that scan is cold in the liver, and you're just seeing physiologic uptake in the heart, kidney, and bladder. But to me, one of the most dramatic findings from the gastrointestinal stromal tumor trials was work done by Jonathan Fletcher, Chris Corliss, and Mike Heinrich in Oregon and Boston. And that was to do mutational analysis of the KIT gene on every single patient who enrolled in the clinical trial. And in patients who had the most common mutation, an intracellular activating mutation in the KIT exon 11, the response rate is 82%. In contrast, patients who expressed wild-type KIT, but no mutations, so they have a target of Imatinib, but it's not mutated, their response rate's 16%. So this to me is a dramatic example that it's not simply expression of the target, it's actually activation or mutation of the target that drove responses to Imatinib in this disease. So what lessons have we learned? For me, the most important lesson is it's the target. And if you have a good target and a good drug, you can get good results. It really can be that simple. Now, I've used this slide for probably close to 12 years. And as you can tell, I put a lot of emphasis on the target. And recently, I've been reminded that we also have to have good drugs. And the example I'd give you are BRAF inhibitors. We have a very good BRAF inhibitor on the market, Vemurafenib, high response rate in melanoma. But I give a lot of credit to Plexicon and ultimately Roche to take that drug through, because there have been 2 examples of RAF inhibitors that were not very good mutant BRAF inhibitors that have been tried in melanoma and they didn't work. And people then began to question, is this really a good target? When I looked at the data, I said these drugs are not very good at shutting down mutant BRAF. We need a good mutant BRAF inhibitor. And when a good mutant BRAF inhibitor came along, we saw a 60 to 80% response rate in patients with mutant BRAF. So it just reminds me that we need not only a good target, but also a good drug to get good results. So if we're going to translate the success of Imatinib to other cancers, we need to start by identifying the right targets. And in my mind, these are going to be the early molecular pathogenic events. We need to treat as early as possible in the course of the disease when our response rate and durability is going to be greater. And for that, we need to develop highly reliable, specific tests for early detection. And lastly, we have to do a much better job of matching the right patient with the right drug. Activated ABL inhibitor, activated KIT inhibitor, and so on and so forth. And in addition, we also, of course, need to understand resistant mechanisms so we can begin to think about using the appropriate combinations of therapy. So I've already mentioned Vemurafenib, but there are already examples of how this paradigm has translated with other kinase inhibitors. So EGF receptor inhibitors in mutated EGF lung cancer with a 60% response rate, Dasatinib, an ALK inhibitor in patients with ALK fusions, a 60% response rate. So in fact, there already are other examples of how this paradigm has translated to other cancers. What I want to finish with is how our lab is trying to accelerate target identification and validation. And when you think about target to therapeutic, and I'll just give modern history in CML, the Philadelphia chromosome discovered in 1960, Janet Rowley's work in 1973, BCR-ABL in the 1980s, and FDA approval of Imatinib in 2001. Overall, that was a 40-year process. And we kind of can break that down into two phases. One was the first 25 years of target identification, and the second 15 years or so was drug discovery and development. So I'm going to talk about what we're doing for decreasing the time for target identification. And when you think about how you can do it, we kind of have these two disparate communities. We have our genomics people who say everything's going to get answered by genomics. And we have the group on the functional side that said, well, we need functional assays they're going to tell us. And what our lab has tried to do is we've tried to say, well, let's try to do both. So we look at genomics, and we can do whole genome RNA-seq, and it's great. We get lots of data, but our problem has been we get lots of passenger mutations, and getting to validating what is the actual driver mutation can be quite difficult and quite time-consuming. Or we can go to functional screens. In our lab, we've used RNAi-based screens, kinase inhibitor screens, and we get lots of targets, but sometimes we don't get mechanism. So what we've now done is we've said, let's combine these two. Let's integrate the results. Let's prioritize genomic data, and let's figure out what's happening in our functional screens and explain that. And I'll take you through a couple of examples of what we're doing in leukemia, combining this type of data. So for our functional screens, we're using an RNAi-based screen. We have a 96-well plate where we knock down each tyrosine kinase individually. And we also have a kinase inhibitor screen where we actually have about 140 inhibitors, a core of 38. They're profiled broadly against almost the entire kinome, and we can use this data to help us identify targets. Our readout in these functional assays is a 3 to 4 day cell viability readout. So here we're looking at leukemias. In our kinase inhibitor screen, acute lymphoblastic leukemia, acute myeloid leukemia, and myeloproliferative neoplasms. In 70% of our samples, we see sensitivity to a kinase inhibitor. But if you look at this heat map and the dark red is sensitive and the open areas are insensitive, what you see is enormous heterogeneity. And no matter what algorithms we use, how we cluster this data, we see lots and lots of heterogeneity. And so just to show you how this data is represented, vertically it's individual patients, horizontally it's individual inhibitors. But again, no matter how we look at this data, we see just enormous heterogeneity. In our RNAi-based kinome screen, again, vertically individual patients, horizontally individual knockdown of genes, again, enormous heterogeneity. Now, as you might expect, we see in asterisks on the far right, we see some JAK2, we see some FLT3, some of the known mutations, but again, lots of heterogeneity. Overall, about 30 to 40% of patients with this individual gene knockdown are sensitive to one individual kinase. And I'll take you through a couple of examples of how we've used this data to identify specific targets. Here's a myeloproliferative neoplasm, chronic myelomonocytic leukemia, sensitive only to JAK2. Each of the blue bars represent an individual well and individual kinase. JAK2 is the only kinase, if we knock down, we see loss of viability. JAK2 should be pretty obvious. Let's go and sequence that. Comes back wild type. So that's our problem. Some of these functional screens don't tell us mechanism. So now we went to a much wider sequencing platform and identified the thrombopoietin receptor, or MPL, which is a known activator of JAK2 with a 2-base pair insertion that alters the last 46 amino acids and puts a new C terminus. And that, in fact, is transforming in a myeloid background. So now we've actually explained mechanism. We've gone from an RNA-based screen, JAK2 sensitivity, driven by a thrombopoietin receptor mutation. Let's go to our inhibitor screen. Do we find any drugs? And it turns out that midostaurin actually has JAK2 inhibitory activity. You can see on the far right, if you stimulate cells with thrombopoietin, we can inhibit the activation of JAK2 with midostaurin. We were fortunate that this patient was able to enroll in a clinical trial at Stanford with Jason Gottlieb. And this patient responded rapidly and dramatically and had a response duration of close to 1 year on midostaurin. So in the space of 3 months, we went from a patient sample, identified a specific mutation, explained the mechanism of action, and got this patient on a clinical trial that worked. So we took 40 years or 25 years down to 3 months. So I'll give you another example of a patient with neutrophilic leukemia. We started with our sequencing data, where we've been sequencing pretty close to whole genome, whole exome. We came up with about 20 high priority mutations, and we tried to analyze them according to pathways. And we really couldn't see any relationship between the mutations we identified. So we then brought in our functional screens. And our functional screens showed a Src family member, FGR figure, and another tyrosine kinase, intracellular kinase called TNK2. So these two kinases seemed to mediate sensitivity in this sample. And we went to our drug screen and saw similar sensitivities with Dasatinib, a potent Src family inhibitor, as well as a TNK2. Now for these drug screens, we've developed algorithms to help us identify what the targets are based on the inhibitor profile. And as it turns out, Src family members and FGR were very high on the list of inhibitor sensitivity. So now we can begin to use this functional data and integrate it with our genomic data, and we can actually come up with the implicated pathway. So in red, we have our siRNA sensitive samples, which were FGR and TNK2 over on the left in red. In the middle, we have all of our drug targets, and on the outside in blue, we've got our mutated genes. And so now we've taken the space of 20 to 50 mutated genes, and now we have 4 high priority mutations for validation. We put these 4 mutations into BAF3 cells, and it turns out that the G-CSF receptor mutation, which is one of our 4 high priority mutations for validation, is activating and transforming. Now, if you did a literature search, you would have said G-CSF receptor should signal through JAK2. As it turns out, our kinase inhibitor screen said we have Src sensitivity. When we put our G-CSF receptor mutations into BAF3, in fact, they predominantly signal through Src family members. So when we think about integrating drug targets, if we had just gone with the genomic data, we would have said we have a CSF3R mutation, and it's likely going to signal through JAK2. If we had just gone with our functional data, we'd have said, well, we've got these Src family hits, but we have no idea why. When we integrate all of this, we come up with G-CSF receptor mutation signaling through JAK2 and some drug targets, and we can do this all in the space of a couple of months. So if we think now about what we need to do in this century with cancer research, I think I've gone through some of these important issues. We have to identify all the molecular pathogenic events in cancer. We have to do much better with diagnostics so we can get early diagnostics, and we need to think about imaging so we can understand more quickly how our therapies are working, who they're working for, and even beginning to think about rational design of combinations. I didn't talk about the drug discovery, but it's clear that we need to compress the timeline of getting drugs out of labs into the clinic. And lastly, when we think into the future, if we're really going to think about preventive cancer treatments, we need to think about that based on our genetic risk of getting cancer and begin to design molecularly targeted therapies that will actually target genetic lesions that predispose people to cancer. But lest you think that I'm all about targeted therapy, I do believe that we have to take a broad-based approach to cancer. Yes, I'd like to see far more targeted therapies directed at critical targets in cancer. But we also have to think about prevention and early diagnosis. And we also have to remember part of the way that we're going to eradicate cancer is also going to be by bringing in immune therapies that can eradicate minimal residual disease. And if we take this broad-based approach to cancer, it's my view that we can make significant impacts against cancer in this century. So in closing, I'd just like to acknowledge all the people at my Cancer Institute who have helped me over the past almost 20 years, all the people throughout the world who put patients on our clinical trials, Novartis who funded the clinical trials, and all of my funding agencies, NCI for many, many years, the Leukemia and Lymphoma Society, and most recently Howard Hughes Medical Institute. But what I really want to thank are the patients who've gone on this journey with me. Patients who had been told to get their affairs in order, patients who are now still here alive and thriving, like this patient, the longest continuous patient on Imatinib in the world, 13 years and 9 months, still continuing to do the things that she enjoys doing, like gardening. Patients who've traveled from Italy and Seattle, who love dancing. Patients who are referred from hospice and are spending time with their grandchildren now 12 years later. Patients traveling from many, many places for our clinical trials to Oregon. And some patients doing truly remarkable things. This was the first patient from Australia who was treated with Imatinib in the year 2000. She traveled back to Australia and was selected as one of the torchbearers in the Sydney Olympic Games in the year 2000. So these extraordinary examples begin to add up. We take our patients one at a time, we take our diseases one at a time, and pretty soon we have large groups of patients, large numbers of diseases, surviving and thriving despite a diagnosis of cancer. And this is the image I want to leave you with in my hope for the future. Thank you very much.